Hi there. Welcome to Mushroom Hour. Today on Mushroom Hour, we are blessed to speak with our very special guest, Alan Rockefeller. Alan is a mycologist who has undertaken extensive field and lab research on the mushrooms of California and Mexico. He has been collecting mushrooms for 17 plus years and has focused on the taxonomy and photography of Mexican mushrooms for the past 11 plus years. Alan has collected mushrooms from the Mexican states of Jalisco, Colima, Michoacan, District Federal, Morelos, Puebla, Veracruz, and Oaxaca. Now, these trips can involve dangerous nighttime excursions into landslide-prone areas, and on the Vice TV show, Hamilton's Pharmacopoeia, Alan was featured hacking through the jungle with a machete, hunting for rare fungi. On what I am now calling daring expeditions, he finds rarities including bioluminescent species, psilocybin-containing species, and other incredible fungi. A fair share of these finds have been mycological specimens that were new to science. Alan is also an active moderator on shroomery.org forums and almost every Wild Mushroom ID Facebook group that I've seen out there. When he's not photographing mushrooms, he is busy sequencing fungal DNA, photographing the microscopic features of mushrooms, hacking computers, modifying electronics, and working in the IT security field. Alan, it's a privilege to have you here. Thank you so much for coming on the show.
Thanks for having me on.
I'm excited to talk about all of the adventures you go on, all the amazing fungi that you find in Mexico. With your knowledge, we could talk about a million different topics, but we'll stay with Mexico and some of the finds there. So before we jump into it, why don't we get just a little insight into Alan, maybe how you got where you are today. So anything about your early life, maybe influences within your family, within academia, or maybe just some synchronicities that aligned to put you in a place where you became obsessed with mushrooms?
I've always been interested in nature. Starting from a really early age, I went camping with my parents and the Boy Scouts and all that. It's always been nice to spend time outside. But before I got into mushrooms, it kind of seemed like I would be wasting my time when I went outside. Like I'd come to the forest and it'd be like, wow, this is beautiful, but what am I doing out here? Finding mushrooms gave me a good reason to go to the forest and a good excuse to go every day. So that's been really nice. And I got into mushrooms when I was hiking. I went hiking on Christmas Day in 2001, and mushroom season was really good. And I was down in Redwood City, California, and there was this mushrooms everywhere. And looking back on it now, I know they were mostly Rusilla and boring stuff like that. But I thought, wow, some of these must be interesting and rare. And so I decided that, there must be people out there that knew how to identify these things, which ones were cool, which ones were poisonous. So I decided to start taking pictures of them, just paying attention to them. And that's how I got where I am.
And that's what I tell a lot of people is if you want to get into the world of mushrooms, just go out and start looking. And it sounds like that's what happened for you. You went out, start noticing mushrooms, start taking pictures. And before you know it, you'll be on the forefront of mycology and discovering new species like Alan maybe. Getting to mushrooms in Mexico, it's a fascinating topic, something I don't know a lot about. I did this past September take a trip down to Oaxaca and was exposed a little bit to the mushroom culture there. It's obviously deeply embedded in the culture of villages surrounding the city when you go up in the mountains there, to the point where they even had a mushroom tourism station where we could sign up and got a guide, and they went out and took us around showing us all the mushrooms that they knew of in the area. But when did your first journeys to Mexico start? What was the impetus to go to Mexico in the first place? What were those first journeys like for you?
Well, that was around 2007. There was a guy on shroomery.org, and his name was Cactu, and he was posting pictures of Psilocybe zapotecorum and Psilocybe cerulescens. And they were much different than the Psilocybes that we have here in North America. They were really big, really cool looking compared to the little ones we have in the United States. So I just sent him a message and asked him if I could come mushroom hunting with him. And I was really surprised when he said, yeah, sure. So I just took vacation from work and booked the plane ticket and landed in Guadalajara. And he ended up being real cool. He was a biologist, even spoke English. And so within an hour of getting off the plane, I was in his Psilocybe cerulescens patch up in La Primavera, which is the forest above Guadalajara. and just went around Jalisco for about a week, and went to a whole bunch of really cool places. And I was pretty much hooked by then. So then 2008, I went back for a couple of weeks to Mexico in 2009. And then 2010, I decided just to quit my job and do it full time. And so usually I'll spend about 5 months per year in Mexico these days.
Yeah, and it's funny hearing that story of this biologist saying, "Yeah, just come on down," because that was a little bit of the experience when I first met you. You started in the context of a Bay Area applied mycology meeting, and you were talking about trips to Mexico, and I remember a number of us were like, "That sounds amazing, Alan. We'd love to go." And you're like, "Yeah, come." And it's
Yeah.
funny how a lot of times people who are into mushrooms, if you're interested, it's not as hard as you think to get involved. And I think a lot of people who go on those explorations like you do, it's nice to have people with you probably.
Yeah, for sure. There's a lot of mushrooms in Mexico for sure. It seems like the rainy seasons there are a little bit more reliable than the rainy seasons in California. The monsoons come in the summer, so Mexican mushroom season will be June through September. There's just like a whole lot of different habitats, a whole lot of different elevations. The plant diversity in Mexico is so much higher than it is in California. So the mushroom diversity follows the plant diversity. And so you'll just find all sorts of cool things.
Right. That gets to the heart of one of the questions I had here. Were there any unique environmental factors about Mexico that make it such a great host to fungi? And I think that explains it beautifully, is the incredible plant diversity you have there. Now, I'd imagine region by region, you see a lot of diversity too.
Yeah, absolutely. And even more so just in different elevations. So, Mexico is very mountainous. So the humidity comes off of the ocean and hits these mountains and drops all the rain there. So they get a lot of rain. But at different elevations, you have very different habitat, especially because the temperature is very different and the amount of rain is very different at different elevations. So you go up or down 100, 200 meters, and you'll be seeing very different plants and very different mushrooms. So the same mountain will have many different habitat zones.
That's something I'm just starting to key into, is the effect of elevation. Obviously on plant populations, you see it, but of course then on fungi populations, elevation would play a huge role as well. Now, in taking these trips to Mexico, are you still in contact with that first gentleman you went with, or who ends up being your support system if you need one when you're there in Mexico? Are there folks that you stay with? Do you need any types of guides in particular when you're down there? Just because, it is a foreign country and it's not guaranteed that you'll be able to navigate it really successfully potentially. So do you have a support system there, a group of people that you kind of connect with when you're down there to help make these trips easier?
Well, the first couple of years I was visiting my friend Cactu, but he moved to Nicaragua in 2011. So by 2011, I already had a lot of my favorite places, a lot of the national parks, a lot of the volcanoes that I went to with him. So usually I'm by myself. I have some friends in some of the different cities, and I'll stay there occasionally, but most days I'm camping out. And, most people in Mexico, they either have to go to school or they have to work. So, most of my friends can hunt for 1 or 2 days, but they can't just take off across the country like I can.
Right. So you're just out there with your tent, your supplies, and you're just kind of legging it between all these different places that all these different sites you want to visit.
Yeah, I drive my car down there. So, it carries all the camping equipment, microscope, DNA sequencing lab.
Wow. So you have the whole thing packed up into your car. That's amazing. One of the fascinating fungi that I know you see down there, and I've actually seen one of your talks at the Marin Mycological Society, was about bioluminescent fungi. These are something that hold a very special fascination to everybody who gets into mushrooms. The idea that you could have something biological glowing in the dark just conjures up all kinds of amazing images. So I guess tell us a little bit about bioluminescent fungi. Maybe we can start with, how do they work? How are there mushrooms that glow in the dark?
Well, there's two different kinds of mushrooms that glow in the dark. You have the bioluminescent mushrooms, which make their own light, and fluorescent mushrooms, which take ultraviolet light and convert it into visible light. So for the fluorescent mushrooms, you walk at night with the black light. It's just like a cheap ultraviolet flashlight. And then certain mushrooms will be glowing in the black light. Those are pretty cool. You get all sorts of different colors coming off of the fluorescent mushrooms. A lot of whites and greens and purples and oranges coming off of there. And then the other type is the bioluminescent mushrooms, and those make their own light. So you walk with no light at all. You'll just see them emitting a green light. And there's a lot of different species. I've seen about 20 different species in Mexico and about 10 in California that glow in the dark. And so to find them, what I'll do is turn off all the lights, and I'll do this on a time when there's no moon. So either a new moon or wait for the moon to set. And then after about 5 minutes, your eyes adjust to the dark, and then you start walking. The other way to do it is just during the day, you collect all the mushrooms that you think might glow, and then look at them either in a dark room or after it gets dark. So usually I'll find like a trail, which is not too treacherous, no cliffs or anything. So I'll kind of walk it in the afternoon. And then if it looks like it's a good candidate for walking at night, then I'll come back after the sun goes down. And if it's near a city, then you have to do it on a very clear night. So all of the light from the city goes straight up into the air and doesn't bounce off the clouds because it needs to be absolutely dark. And when it's not absolutely dark, any light-colored thing, a light-colored piece of bark or a piece of garbage will look like it's glowing in the moonlight. But when it's extremely dark, the only thing that you see are the things that glow, and that's mostly mushrooms and insects. So the insects you can always tell because they make a little pinpoint of light. And sometimes it's 1 dot, sometimes it's 2 dots, sometimes it's a row of 16 dots, but it's a very small source of light. So whenever I see something like that, I know it's a bug. And I'll look at it for a second, but mostly ignore it. Mushrooms, by contrast, they are a much more spread out source of light. So it'll be the whole cap of the mushroom may be glowing, or just the stem, or the cap, stem, and mycelium, sometimes just the mycelium. So if it's just the stem, you'll see like a vertical line glowing in the forest at night.
Right.
Or, the caps, obviously just look like glowing mushroom caps, little glowing circles. And sometimes there's just one, sometimes there's thousands of them. So it can look like a whole bunch of stars in the night on the forest floor. And then sometimes the mycelium glows. So sometimes there will be just, beds of leaves, and every leaf will be glowing. So you could be walking through the woods at night.
Wow.
10,000 glowing leaves, and it's just like an ocean of green light coming up from the floor. They're not evenly distributed. They're in certain spots, so you'll be walking for a while and not see any, and then maybe you'll see a whole bunch in one spot. They're usually in the wetter areas, very often near a stream, near water, or just anywhere that the humidity collects. And they're all different brightnesses. So you get some that are extremely dim, and some of them that are very bright. So the brightest one, I found it in 2014 in the state of Hidalgo, cloud forest there. And that one has been named Mycena perlae. It's really bright. Only the cap glows, and it's so bright that if you just pick one up in the middle of the day and put it up to your eye, you can see the green light coming off of it. And if you throw it at night, it just looks like a shooting star. So when I take a picture of that, I can turn the ISO on the camera down, and exposure is only about 6 seconds or so to get a real nice picture of it. Whereas some of them are extremely dim, and you'll just barely see them glowing. And sometimes I'll do a much longer exposure, like a 40-minute exposure, to capture enough light to take a good picture of those glowing.
I mean, that sounds magical, seeing these 10,000 leaves all lit up with some kind of mushroom mycelium. So the area tends to be these wetter areas near rivers or streams. So that's kind of how you're keying into where you might find them. But during the day, are there any properties of the fruit bodies of these mushrooms that, if they're not the Mycena perlae where you can see it even during the day, are there any indications or common features of these mushrooms that might give you a hint that they might be bioluminescent?
Yeah, absolutely. About 95% of the bioluminescent mushrooms are Mycena. So mycenas are pretty easy to identify because they're growing on wood or leaves, and they got white spores, and they're small with a thin stem. They all look different with all the different colors and textures, but they all look very similar as well. So you can just look at a mushroom and know it's a mycena. And so what I'll do is I'll just collect all the mycenas, take nice pictures of them in habitat, and put them in a tackle box. And then later I'll just see which compartments of the tackle box are glowing. And usually the way I'll do that is when I get into a completely dark area, I'll turn the ISO on my camera all the way up. So it's like ISO 200,000 and do a 30-second exposure. So with maximum sensitivity, then, it's really easy to see which compartments in the tackle box are glowing, but you get a lot of digital noise, that sort of ISO. So that's just the first pass to see which ones are glowing. And then when I find the ones that are glowing, I'll set them up in a nice aesthetic way and turn the ISO way down, maybe more like ISO 1600, and do an exposure for more like 10 minutes. And that's when I'll get the actual good quality pictures that are worth publishing.
It's interesting to hear that actually in situ they won't be glowing with power like that, but it actually requires a process of exposure to really bring through that glow. And it actually gets to the fact that, yeah, you probably have to walk in this absolute pitch black to be able to see these things. And I'm sure over the years your eyes have developed to be keenly aware of any differences in light.
Oh,
So
I think my eyes are the same, but certainly how I process what comes in from the eyes has changed a lot over the years. It used to be I would stop and look at every glowing bug. And now, I pretty much know what's going to be a mushroom from longer away. And it is pretty dangerous walking at night. I don't really run into snakes or anything harmful like that. But sometimes there's, holes or streams or cliffs or branches that like to poke you in the eye. So it's a real good idea to wear glasses. Not only does it make everything a little bit sharper, but it'll stop sticks from poking you in the eye.
yeah, you don't want a little stick in the eye when you're out looking for glowing fungi.
Yeah, I mean, it's not a big problem because when you're walking at night with no light, you're walking extremely slowly. So it's not like you're running through the woods or anything like that. It's like you take a step and kind of look around and take another step. And, maybe you go 100 yards in maybe 15 minutes.
I don't think running through the woods looking for these mushrooms is going to yield many results. Now, if you know about that underlying process, what is the process that makes these things glow or bioluminesce? And are there any different colors that these things can glow?
There's no different colors. Everything on Earth that's not in the oceans but out in the air, it glows at the same color. Except for some glowworms in New Zealand, some caves in New Zealand, they have a blue glow. But everything else on land will have a green glow that's between 520 and 530 nanometers. So that indicates that it's the same chemical system, which has actually evolved several different times, but it's the same sorts of chemicals reacting. And Brian Perry just gave a really good talk. I posted it on my Facebook timeline where he dives into the chemistry of what exactly is happening. But you have luciferin and luciferase. These are enzymes made using a acid called caffeic acid, which is present in pretty much all plants. But if you're growing these things in culture, a lot of times you have to add caffeic acid, which you can get on eBay. And that makes your petri dishes glow a lot brighter. It makes your grain jars glow a whole lot brighter if you're doing like grain spawn of Pinella stypticus or omphaliotis or anything like that. But the luciferin reacts with the luciferase, and that releases carbon dioxide and a photon of light. Very similar to a glow stick, but different chemicals involved. And then the mushrooms can recycle those enzymes to turn them back into the luciferin and release more light. So it doesn't take a whole lot of energy for the mushrooms to create the light.
And it sounds like a lot of other organisms have developed a similar process when you think of bioluminescence to generate that same 520 to 530 nanometer light wave. Those instances of convergent evolution is always really fascinating, especially when it makes something glow in the dark. In terms of cultivation, that was going to be one of my questions. How do we all grow our own glow in the dark mushrooms? Are there any species that are being cultivated and used by people for these glow in the dark properties? Are there any that DIY mycologists can grow themselves?
There's two that you can buy. If you just Google for it, you can find plenty of vendors selling liquid culture syringes, and that's Pinella stypticus, which really should be a mycena, but it's kind of like a little polypore type thing. And then you also have Omphaliotis olivescens, which is native to Australia, and it's looks a lot like an oyster mushroom, but it can be kind of bluish. So that's the poisonous thing that not as many people grow. So the most common one is Pinella stypticus, because it's not poisonous. And it glows pretty well. It really likes acidic conditions. So when I grow it, I'll put a little bit of citric acid in the auger or in with the grain, and it colonizes a lot faster, is brighter and much happier when it's kind of down around pH 4. But yeah, you can just make a bag with grain and sawdust. What my friend usually does, sawdust supplemented with rice bran, a little bit of citric acid, and he'll put that in a filter patch bag and inject it with a liquid culture. And liquid culture is available from Sporeworks or a bunch of other vendors. He gave me a bag one time, and it was glowing for about a year and a half.
Wow.
So the mycelium itself glows. Then if you fruit it, it gets about 3 times brighter. So you can just keep it in the bag and just use it as a light that way. Or you can fruit it. You could make plug spawn with it and grow it on logs. I see it out in the field a lot. People say that on the West Coast it doesn't glow, and on the East Coast it does. But I found some real nice glowing ones in Sonoma County a couple months ago. But yeah, you can certainly just take a tan oak log and drill holes in it and stuff that sawdust or make plug spawn and make glowing logs that way. Now, the glowing is not super bright on this species, so if you're going to make a trail that's lit with glowing mushrooms, you're going to have to be pretty far away from the city and have your eyes adjusted to the dark to make that doable. But if you live out in the country, then you could certainly do something like that.
You just called out the mental image I had, which is of your own glowing mushroom lights, around your property. It doesn't sound like to grow, at least the Pinella stypticus, there isn't a crazy, strange substrate or food source that you need. I mean, it sounds like things that everyone could get their hands on and start growing this. That's really inspiring. It makes me want to start growing my own glow-in-the-dark mushrooms. Are there any other applications people are starting to see for bioluminescent fungi?
Well, those ones that are commercially available are not very bright. And there's about 10 other species in California that are even dimmer. I've recently been taking a lot of pictures of those. But there's a couple that are really bright. The Mycena perle and Neonothopanus gardneri are both very bright. And so just about a week ago, a paper came out where they took the Neonothopanus, sequenced the genome, and took those genes out of the mushroom and put them into plants. And so now they have glowing tobacco plants. And those work pretty well. I kind of doubt they'll really be bright enough to replace LEDs or anything like that. More of a novelty, and there probably always will be, but it's certainly possible to make plants that glow now.
Wow, so they're actually taking out that part of the genome and being able to put it into something else. Maybe one day we'll be putting that in people or something and making ourselves glow in the dark.
Yeah, you certainly could. Might be some ethical issues with that, but certainly glowing pets are possible.
Glowing pets are now possible. And as you've done this work, I believe a number of the bioluminescent species you found in Mexico were actually the first time they had been found and cataloged. Is that right?
Yeah, there was a bunch of them that had never been seen before. So about a year ago, I published a paper with some co-authors that named 6 new species from Mexico. And there's a lot that we've found, maybe 20, 25 of them or so. Most of them are really rare, and I've only seen them a couple times. To really name a new species, you don't want to just name it off of one collection. You want to find it a few times so you know the range of macroscopic and microscopic and different habitats that it can occur in. Get real familiar with it before you name it. But yeah, those 6 are pretty common, and so those have been named. And then there's a bunch more that are being worked on that are more rare. So yeah, there's definitely a lot to discover there.
Well, and for those of us who haven't ever discovered a species of fungi, what is that process like to get it added into the mycological lexicon? I mean, what level of information you need? It sounds like you need to find it multiple times, first of all, with multiple specimens, but what are kind of the next steps in getting something codified and added to the library of known fungi?
So you want to write a good description of it, both macroscopic and microscopic, and sequence the DNA and upload the DNA sequences to GenBank, and then write a paper, ideally a peer-reviewed paper that gets published. And once that gets published, then that's the official name forever.
Wow. And so you can pick out whatever name you want.
Has to follow the rules, has to be either Latin or Greek. And it's really best to name something after a unique feature that separates it from its closest relatives. Because if you just name it after your friend, in 100,000 years, people will be like, well, who is that? That doesn't have much meaning. So a name that separates it from the close relatives is going to be still meaningful in 100,000 years.
Yeah, you do want to leave a legacy that's going to make sense to people down the road, not name everything after yourself or your pet or that kind
Well, you
of thing.
can't name it after yourself, but you can name it after anything other than yourself.
It kind of curbs a little bit of the egoism then in the naming of the mushroom.
Yeah, they made that rule.
Now, do we have any information, because you've studied this, do we have any information about the evolutionary history of bioluminescence? Maybe why mushrooms would develop bioluminescence? Is there any evolutionary relevance we found there?
Well, mushrooms certainly benefit a lot from attracting animals and insects because, they drop their spores and they try to get the wind to disperse the spores, but most of the spores fall within just a couple feet of the mushroom, so the wind is usually not very effective for that. And then when the wind does disperse the spores further, you need two spores to germinate right next to each other to make the dikaryotic mycelium. So it's not as effective as it can be if you have some sort of animal that wants to eat the mushroom or walk on the mushroom, gets attracted to it, and then drags big clumps of the spores or mycelium to a different substrate that's not colonized yet somewhere else in the forest. So nobody knows for sure why they glow, and there might not be a reason, but that's the reason that rings true most to me. It could also be that they just need to get rid of oxygen, so this releasing the photon is a way for them to get rid of extra oxygen, kind of like an antioxidant. Or maybe it's just completely random. A lot of stuff in nature doesn't have a reason, doesn't have a purpose. And that's fine. It doesn't need to have a purpose as long as it's not too expensive for the organism to create these chemicals. So it's not expending too much energy to create these chemicals, and it's not going to be selected against in nature. So it could just be random.
Could just be an offshoot of nature's endless diversity, but I can't help but try to reconcile some reason for it. And I think that reason does seem probably the most sensible to me, is that it's trying to attract things at night, if there is any kind of purpose, trying to attract something to eat it and spread the spores. Have you had any overlap in your research with academics, maybe in Mexico? Are there a lot of other people researching bioluminescent fungi, and have you been able to work with them in furthering that research in any way?
I think the main guy in Mexico that's doing bioluminescent fungi is Alonso Cortez Perez, and I've certainly found a bunch with him. In the United States, it's mostly Brian Perry and Dennis Desjardins. They're more academic types working at universities, so I'll see them at mushroom events and stuff. I don't really go out mushroom hunting with them, but I certainly talk to them at events and stuff like that.
And that's really important to highlight, that I always love to share with people, is the role of the amateur, someone who doesn't have a PhD or an academic background necessarily, the role that someone who just loves mushrooms and gets out in the field, the impact they can have on the field of mycology. And I think you're an amazing example of that, the fact that you are contributing to the work of various academics like the folks you just mentioned, when your background really is falling in love with it out in the woods back in 2001. I mean, is mycology, because it's a new science, just unique in the ability for amateurs and people who fall in love with it and decide to get out in the field, citizen scientists? Is it unique in its ability for citizen scientists to have a major contribution?
It's certainly easier for citizen scientists in mycology than it is in other fields. Most of the plants are discovered, though there are amateur botanists that keep discovering new plants by paying close attention to the flowers and stuff like that. But yeah, probably, almost half the mushrooms in California are not discovered yet. Even some of the very common ones that you'll buy in supermarkets still have no scientific name. So there's certainly a lot to be that can be done. Just mushroom hunters can make a big impact by taking pictures of the mushrooms and uploading them to websites like iNaturalist and Mushroom Observer. So with iNaturalist, it's an app for Android or iPhone, and you can just take a picture of any living thing, plants, trees, mushrooms, whatever. And as soon as you get cell phone service, it automatically all uploads to the database, and then people will identify it for you. It also has artificial intelligence to try to figure out what it is. And then Mushroom Observer is a website. And so Mushroom Observer just focuses on mushrooms. So on Mushroom Observer, you're much more likely to get your mushrooms identified because the people that visit Mushroom Observer tend to be more serious mushroom people, whereas the iNaturalist people that use iNaturalist are more often people that just like nature and like hiking, but snap pictures of all the mushrooms and plants and stuff they see. So they're both really good websites, and I spend usually like an hour a day identifying stuff on these websites. And so what I do right now is I'll identify everything that gets uploaded to Mushroom Observer worldwide. And then on iNaturalist, there's just too much unless I wanted to do it 8 hours a day. So on iNaturalist, I'll identify everything that gets uploaded in the Bay Area, California, or whichever region I'm in. So I'm doing Oregon these days. And also there's regions I'm interested in. So I'm also identifying everything on iNaturalist that comes across for Mexico and for the Philadelphia area.
Yeah, I'm hearing so much about iNaturalist from different guests I'm having on and how important it is to the exploration of biodiversity right now, both in people being able to contribute their finds. I just had one guest on who said the most important thing you can do to contribute to citizen science as an amateur mushroom hunter is share your find. Don't let that find stop with you. So it's important in that aspect. And then it's also such a useful tool as basically a phycology report where you can get an idea of what's in a certain area just by searching iNaturalist. Just over the past couple months, I made a promise to myself that I'm going to integrate using iNaturalist into my mushroom hunts to make them feel like they're contributing a little more by sharing those finds, and it'll probably help me find some new stuff as well. And I'm always stunned. You just talked about your being able to contribute and identify. I'm always stunned by how active you are on some of these online forums in identifying and helping people figure out what mushrooms they have. I guess what has that online community meant to you? Because you are on shroomery.org, you're on all these different Facebooks, iNaturalist, Mushroom Observer. What has being part of that online community meant to you?
Well, a lot of it is how I started to identify mushrooms and learn to identify mushrooms. And before all these communities, I would mostly learn by editing Wikipedia. So I edited a lot of the Wikipedia articles and started a lot of Wikipedia articles on mushrooms. And then around 2007, I noticed the Shroomery. Shortly after that, Facebook. So I started identifying stuff on there. And when I started, it would take me about half an hour to identify each mushroom. So somebody would post some mushroom pictures, and I would crack open the books and go to the key and just key them out. And after a while, I knew which parts of the key to go to. So instead of half an hour, it was like 5 minutes. Just kept doing it. And, it's a really good way to see what's coming up in different parts of the world, where the mushroom season is good, and just practice all those scientific names and just, get it, lots of repetition. And also I'll just help people out. I can do my good deed for the day, just sitting down at the computer and typing for a few minutes and identifying all the mushrooms for people. So I've gotten, really good at just putting names on stuff. Now I see a mushroom and within about a third of a second, I know what it is. So just writing down and driving along the freeway, I know what all the mushrooms on the side of the road are. And I can go on sites like iNaturalist and Mushroom Observer and the Facebook identification forums. And just, real quick, I'll see a mushroom, I'll Google it to get the correct spelling and a good website link, usually Wikipedia or MycoWeb or Mushroom Expert, and paste that link. So now it only takes a couple seconds to identify a mushroom. And so I think it's a pretty good use of my time when I can spend just a couple seconds telling people what they have, whereas it would take them, a long time to try to figure it out on their own, and they might not even be right a lot of the time. It seems like a pretty good thing to do, just to go on there and help people out, and helps me at the same time just by practicing. And occasionally something really rare will turn up on these sites. And one of the most important things about these sites is that you can generate maps, like distribution maps of where things are seen. So both on Mushroom Observer and iNaturalist, if you're interested in a certain taxon, you can search for it and see where it is. So sometimes it's really exciting if you find it outside of its known range, maybe the furthest south or the furthest west of any time that this mushroom has been spotted. Or if you want to find a certain mushroom, you can just see where it's really common and go to that area. See where in the world it's really common, and also the habitat, what it's growing on, and what sort of spots that it looks like it likes to fruit in. And, adding these biodiversity records really helps, because when someone describes a new species, they can look on there to see where it's found, and they can see all the different photos of it, hundreds of different collections, all the different morphological variations that it can have. That helps a lot, writing descriptions. And it keeps a permanent record. So years later, if you're like, oh, what's that bolete that I found down in Redwood City, you can real easily search and pull it up Usually people are always losing their photos, the hard drives get crashed, laptops get lost. But once you upload it to one of these sites, it's there forever and it'll still be there in 1,000 years. So when people are doing research on global warming or how the habitats change over time, they can see where things were a long time ago and where they are now. So yeah, it's really important to create all these biodiversity records. And it's also just cool from a personal perspective. You can just real quick be like, how many different species of mushrooms have I found? And if I go into Mushroom Observer, it'll say like I found 1,500 species and 400 genera or something like that. It gives an incentive to go out there and find new different stuff and try to get as many different things as you can onto your life list.
And we're at a unique time where these databases, these websites are more eminently available than ever, but they're not brand new. So there's plenty of information already on there to use. So it's like this perfect time to integrate into these biodiversity sites in your own forage practice. That's something I really want to encourage people to do is use these sites. You'll be able to contribute your finds, like you said, keep them for history, and also you'll be able to learn so much. And I love your idea of trying to identify other people's mushroom finds as a good training for yourself. And, that might be something I try to improve my own identification skills, is just have a ton of at-bats, as it were, to try to look something up. And when you're talking about ID keys, can you tell us just a quick overview of an ID key and how it might be different than some mushroom books where you have a picture and a description and you're more trying to identify it that way? What is an ID key and why is it so useful and something people should learn about?
So a key is a text that kind of guides you to different parts of the key depending on what you're seeing. So it'll send you to one page if you have gills, different page if there's pores, different page if there's teeth. And then you'll start going through, the different colors, different textures, and until finally it'll tell you what genus you have. And then the key to that genus will be able to tell you the species. You can just run any mushroom through it from start to finish. And if you pick the right things, you'll get a good ID. So they're pretty useful for that. And also when you're reading through the key, once you think you've keyed it out, you kind of see the other mushrooms that have very similar features in that key. So you can figure out which one of these similar mushrooms you have.
I find it lands you in a place of a lot more specificity much more quickly in using ID keys than a book that, has pictures and where you're trying to key into the physical features. I think the key kind of runs you through this really important mental exercise to look at things you might not have otherwise looked at and guides you a little bit better, but it does take some practice to get used to. Is there any source for ID keys that you use that you find a little bit more useful than others? Is there one go-to for you?
A lot of times, if you just Google for a genus and then the keys will come up for it. It'll be either in a scientific paper or a monograph, or the Pacific Northwest Key Council website has a lot of keys for the West Coast. Just, finding these keys is through Google is a good way to do it.
Like everything else, the internet is one of the best resources.
Yeah, certainly better than any book for mushroom identification.
That's important to know because I used to ask everybody about what their favorite ID books are, and I'm hearing more and more that the internet is really the best resource because obviously it's the compendium of human knowledge, so it's going to be more expansive than any book.
Yeah, there's a few good books out there for the West Coast. Just on the coast, you have Mushrooms of the Redwood Coast, which is probably the world's best ID book. It works really well from like Los Angeles up to Washington. Really made just for California and really on the coastal stuff. But that's a really good book with nice pictures. And then there's some really good books that came out for the East Coast and the Southeast recently. So the best book is certainly dependent on where in the world you're going to be hunting, though most parts of the world don't have any good ID books yet.
Right. Mushrooms of the Redwood Coast is definitely my Bible because I'm here in Marin County, but that is a good point that the regionality of the book is probably the most important. Well, just to cover another massive area of fungi that I know you've explored in Mexico, that is the genus Psilocybe. And it's interesting to me because I don't think that, and you can correct me if I'm wrong, but I don't think your interest in Psilocybe stems from having a psychedelic experience. So what is your interest in psilocybe? Because you've done such a huge amount of work in identifying psilocybe species and finding them, and almost most importantly, photographing and sharing them. What is your interest in psilocybe? Why does it hold such a special fascination for you?
Well, there's not too many people studying them. A lot of people can't study them too well because they have a job and they can't break any laws, and the licenses to work with them can be real hard to get. So for such a genus with so much interest, there's not very many people working on it, just a few people in the world. They also grow in really neat habitats. A lot of times what I'll do is I'll look into the literature to see where a certain psilocybe species has been found, and then I'll go to that place, and while I'm there, I will search for every mushroom that's in that habitat. And, they need a lot of humidity. They're not just out in the desert. They're not just in random places. They're in very special places. So searching for psilocybe definitely brings me to a lot of very unique spots that I never would have thought to go to if I was looking for edibles or, really anything else.
You mentioned that there were only a few people studying them. One person I want to highlight, because he is so important in the study of psilocybe, I didn't even know about him until I started hearing more of your work, hearing your talks. Who was Gaston Guzman? He's this eminent figure in the study of psilocybe in Mexico. But can you tell us a little bit more about him and why we should know about him if people are interested in psilocybe?
Well, Gaston was an academic mycologist. He was working in Mexico City until about 1980, and then went to Jalapa, Veracruz to work at the Institute of Ecology. He described about half of the psilocybe species out there. So he started in the late 50s, and when people found unknown psilocybe species, they would send them to him, and he would name them if they weren't named yet. So that way he became kind of central in the study of psilocybe, and he wrote the monograph, the genus Psilocybe, published in the late 80s. He's definitely was an interesting guy. I only met him a few times. Sometimes I'll go to Institute of Ecology and hang out a little bit in his office and talk about stuff. He's very academic, kind of a straight-laced guy, I would say. I think he only ate them maybe twice. But yeah, he definitely found a lot of mushrooms.
That's amazing that you actually had the opportunity to meet him. And I believe you've also been able to work with his daughter, who's into this same field, Laura Guzman.
Yeah, Laura teaches at the University of Guadalajara in Zapopan, and she's a really great teacher. Her students just love her because she's very patient and very precise and really good at explaining stuff. So it seems like a huge portion of the people that are into mushrooms in Mexico were her student at one time. And she's been really helpful to me. Like when I have questions, she'll answer her emails. Sometimes I'll go and study the mushrooms in the herbarium at the University of Guadalajara. And so she's like super helpful and very giving with her time. Yeah, so she's doing really good work as well.
And I just want to highlight that you've gotten to have these amazing connections with people and contribute this way. It seems like just by showing up and getting out in the field and cataloging and sharing your finds. I mean, were there any special circumstances that led you to being able to meet and work with Gaston and Laura Guzman? Or was it just a matter of coincidence because you decided you were going to work on this and kept showing up and generating relevant findings? I mean, was there any magic there, or was it just your interest pushed you into these circles?
Oh no, there's really nothing special about me. I never went to college for anything. A lot of people say that the most important part of success is just showing up. Yeah. And that's definitely true. Going to the forest often and paying a lot of attention to what you find, and not just looking at the mushrooms, but trying to get real nice pictures of them and saving the dried collections for later study is really important. But as far as meeting these people, usually I'll meet them at fungus events, and then sometimes they'll invite me to give a talk at the university, teach a class with their students, stuff like that. But certainly all of that comes from just showing up.
Yeah, that's a great message to get out there, because love for people who are getting into mushrooms to realize that if you really pursue this and decide to study it and get out in the field and show up and network with people at mycology events and also network with people on the internet, you'll find yourself in a place where you're talking to people that are people you may have once aspired to be, and here you are with them. I mean, it's really a great message to get out there to people. Now, we heard about the hunt for bioluminescent fungi in the middle of the night. Can you tell us a little bit about the hunt for psilocybe there in Mexico? We heard a little bit about the habitat, but what is the actual process of hunting for psilocybe? What does that look like?
So Mexico has more psilocybe than any other country in the world. Maybe 16 or 18 different valid species occur there. The United States is close behind. I think we have maybe 13 in the United States, but we're much larger than Mexico. Right. So, these species mostly occur in the cloud forests. That's where the vast majority of them are. And some of them are in disturbed habitats and others are way out in the middle of nowhere. So one group that's real famous are the derumbes, and that means landslide in Spanish. So they occur in landslides and also in these places where humans have disturbed the soil, like where they built a new road or near a river where the river disturbs the soil. And so to find those, pretty much drive around or use Google Earth to find landslides and then search usually near the bottom of the landslide. Often there'll be water at the bottom of the landslide, which helps. And so it's some of the most dangerous mushroom hunting you can do because these landslides are very steep and very unstable. And that's what creates the special habitat that they need. The Mexican government puts up signs that say Zona de Derumbes, in these landslide zones. So that's often a key to if you're in the right habitat. And the right habitat's usually cloud forests. So these forests are some of the most biodiverse places on the planet as far as fungi and plants go. They get a lot of rain. They're kind of middle elevation. So these places are not tropical. They start at about 1,200 meters elevation. They go up to about 2,600 meters elevation. So if you go up above the cloud forests, it gets much more temperate. You get snow and ice up there. And the mushrooms are real big because big mushrooms work better in colder environments. And then you go down into the cloud forest and it gets much more diverse. The mushrooms are a lot smaller. And then if you go down below that, it gets real hot. So in these hot areas, the real tiny mushrooms work the best. So you see a lot of little white spored stuff, a lot of Mycenia and marasmias. So the middle elevations are always the most interesting. And usually about 3 p.m. every day, the clouds come in and you'll be able to see there's lots of rain there, really regular rain. They're really neat habitats. So in the high elevation, you have Psilocybe millericula and Psilocybe aztecorum, and they like the cold from about, millericula is 2,200 meters up to 3,500 meters, and then the aztecorum is about 2,900 up to 3,500 meters. So those grow just at the tops of the volcanoes in Mexico. And then down in the cloud forest, you'll have a lot of Psilocybe zapotecorum, Ceruleans, Neohalopensis, Hymeae, Fuchsugeni, and several other ones. And then you also have the other habitat you see them a lot in is pastures. So usually pastures where horse or sheep graze, you'll find Psilocybe mexicana, and those grow directly from the grass. And then you also have Psilocybe cubensis and Paniolacinus, and those grow directly from the manure of these ruminant animals. They're not native to Mexico or North America. They came over from Asia because about 500 years ago, we didn't have any of those cows or horses that create the substrate for Psilocybe cubensis and Paniolacinus. They came over with the animals, and now they're naturalized. So those are pretty common. So usually when people go pick magic mushrooms, they go to the cow fields, the horse pastures, and they pick them there. Though really only a very small percentage of the psilocybin mushrooms occur in the dung habitat. Almost none in California. Most of the species you'll find just out in the forest. The derumbes in the disturbed areas, and then others like the Neohalopensis and Hymeae and Fagicula, they occur way in the middle of nowhere where there's no disturbance, usually from little mounds of moss that you get in the cloud forests. There's really a lot of different habitats that you see these things in. And one that I've been looking for about 8 years and finally found this year is Psilocybe moseri. And Psilocybe moseri looks a lot like Psilocybe millericula. And the millericula grows at very high elevation, whereas Psilocybe moseri is tropical, that grows at much lower elevations. And it was discovered in the state of Chiapas. And I visited the type location in Ocosacuautla, Chiapas, several times looking for Psilocybe moseri, but I never found it there. But this February, I went to Ecuador, and there was a really beautiful waterfall, kind of a touristy area, but on the edge of the waterfall, almost behind it, there was a bunch of Psilocybe moseri growing. So I was excited to finally get good pictures of that.
Yeah, was there anything in particular about the moseri that made you really want to find it, or was it just the fact that you hadn't found it before?
Just that I hadn't found it before. Of the species in Mexico, I have really good photos of about 90% of them. So I think the only one that I know of that's left to photograph is Psilocybe meridionalis. And that's known from Sierra de Cacoma in Jalisco. And that's only been found one time, so it's just known from one collection. And so I went and found the type collection in the herbarium and got good pictures of the dried specimens, and also the notes that are along with it give the GPS coordinates of where it was found. And so I've been looking for it even in the exact spot it was found, and I haven't seen it yet, but we really just have to wait for a time where the season's really good, where there's a lot of rain, and then go to the right spot after it's been raining a lot, and it'll be out there.
Yeah, now have you discovered any new species of Psilocybe in Mexico, or are you mainly finding ones that have already been described? Have you discovered any new species of Psilocybe mushrooms in Mexico?
I found one in Veracruz, and this one, it looks sort of like Psilocybe neohalopensis, but the microscopic features are much different. It has these huge chylocystidea that are bifurcating, trifurcating, and so it's definitely nothing that's known before. And I only found one mushroom, so that one mushroom is now in the herbarium in the Institute of Ecology in Jalapa. So I think it wouldn't be a good idea to name a new species just after one mushroom. But it's certainly now we know where it occurs. And so I visit that spot every year to see if I can find it. And I haven't seen it again. So it's pretty rare. It grows always near water in the Jalapa area.
As we're talking about Psilocybe, I again wonder about any evolutionary significance. And I've heard people have a lot of different opinions on this. Is there any evolutionary significance to containing psilocin or psilocybin in the mushroom? As someone who has an intimate relationship with these mushrooms, have you been able to glean any insight about any evolutionary significance there for the life form?
Nobody knows, and my guess is nobody will ever know. You know, it's possible that they're trying to deter predation, but for mushrooms, it's not really an advantage. They usually want to attract animals that like to feed on them.
Right.
That moves the spores around. It's possible they use them as a neurotransmitter, but it seems like the other mushrooms that don't have psilocybin do just as well as far as that goes. It could be that it had some function millions of years ago and doesn't have a function anymore, but since it's not too expensive for the mushroom to produce the chemicals, that they're pretty common tryptamine alkaloids that are made from these really common amino acid backbones. So maybe they don't have a function anymore, or maybe they have a function that we will never know about, or maybe they have a function that sometime in the future we will discover.
That's an interesting idea when you look at nature's diversity. The idea that a function or a feature, if it doesn't consume too much energy and threaten the species' ability to reproduce and maintain its ecological niche, then it'll just keep that unique trait or feature even if it no longer has a function. The idea that these traits like bioluminescence and containing what are to us psychoactive substances might not have any purpose, it's just an output of millions of species of diversity, is pretty awe-invoking as well, to realize that there may not be a reason, but nature is just producing such abundance of variation that this is just one of the things that happens.
Yeah, the evolution is extremely random. You know, the traits that turn out to be beneficial stay, but if it's not beneficial but not too costly either, that can also stay.
Yeah. Now, do you have any guidance in general for people, as us amateur foragers and mushroom hunters, ways that we can capture and catalog our finds that may lend them more to scientific study or be able to contribute the furtherance of mycology? Are there any tips you have for amateur foragers in that vein, maybe methods of collecting or photography that make the find more useful, let's say?
Yeah, absolutely. The most important thing you can do is get good pictures of a mushroom. And the way I learned to take good pictures of a mushroom is 10 years of taking bad pictures of mushrooms. And that was absolutely the wrong way to go about it. because all of my photos that I took before 2010 are completely useless. So if you just kind of like walk up to a mushroom and take a picture, only 1 in 10,000 will actually be good pictures. So it takes a little bit of thought, and it's not hard to take good pictures of mushrooms. I could show somebody how to take really good pictures of mushrooms in just an hour or two. But I think I'm going to start doing that, giving more talks on photography and that sort of thing. Basically, these days, even the newer cell phones take really nice pictures. So you don't have to carry around an expensive camera, though you can certainly get better results if you really know how to use a good camera. So I usually, use a Nikon with a tripod and a macro lens. The most important thing is to find good mushrooms. So if it's real dry out, you're not going to find anything worth taking pictures of. So you need to go when it's been raining. And it doesn't really matter how many days after rain, just as long as the ground's still kind of wet, there'll be good mushrooms out there. Just finding one mushroom, unless it's something really rare, I won't photograph it, because you need a few mushrooms in a few different stages of development to get a good picture. And they're not usually all in the same area. So what you need to do is find a few of them, and sometimes they're kind of spread out. So if I find one, I'll put it in my tackle box. And then at the end of the day, if I haven't found any others, I'll just throw that away. But if I do find others, then I will put them all up, set them all up near each other. And so what I like to do is find a few that are in the ground just naturally, like a nice little cluster, or a few scattered around, and then take a few other ones from the area nearby and lay those down in front of the other ones. So that way you can see all of the features the mushroom has from one photograph. So you can see how it is when it's really young, how the gills change color as the spores mature, how the veil does or doesn't drop, and how the whole thing changes color and texture as it gets older. And so if you put a bunch of mushrooms in different stages of development down in one area, you'll be able to take a picture that's just shows everything you need to know just in one photograph. And that takes a little bit of practice, but with a little bit of thinking, it's really not rocket science. It's pretty easy. You just grab a few mushrooms and throw them on the ground and press the button. And then, as far as taking the picture, just a cell phone can take real nice pictures these days. But what I usually do is I will set up a tripod, and the tripod allows me to use very low ISO. So with a DSLR camera, you can set the ISO. And if you turn the ISO all the way down, you'll get really nice, high-quality, smooth pictures. Even if you zoom in all the way, it'll be really smooth. So by setting the ISO low, you get a real good picture, but that means that your shutter speed is going to be real low. Like in a dark forest, you'll have to have the shutter open for at least a couple seconds. And so that makes a tripod really critical. And also, I'll take a picture, I'll look at my picture and see if it's the way I want it. Maybe there'll be pine needles in front of some of the mushrooms. Maybe some of them won't be in focus. Maybe I'll want to adjust them so they look nicer. So you can just keep taking the same picture until it's perfect. And then the other major thing that you get with one of these DSLR cameras is you can control the depth of field. And the depth of field is how much of the image is in focus. So if you open up the aperture all the way, then the depth of field is very narrow, and it's only a couple inches. So if your mushrooms are spread out more than a couple inches, some of the close ones will be blurry, maybe the far ones are blurry, and you definitely don't want that. By setting up a tripod, and then you close down the aperture to maybe f16, f22, f32 if you really need to, then you can get much wider depth of field. So both close mushrooms and far mushrooms are all in focus. And also, if you just have like one mushroom that you really want to stand out from the background, maybe you want to open up the aperture to f5.6 or something, so the background is really blurred and the foreground is blurred, and then what you want the viewer to focus on really stands out. And then lighting is really important too, because if you just take a picture of the mushroom, the cap will cast a really sharp shadow on the stem. So complete sunlight is always terrible. You never want to take a picture of a mushroom in complete sunlight. If the mushroom is in the sun, then I'll either have a jacket or a backpack blocking the sun, or I'll have a friend stand in the way of the sunlight so there's no direct sunlight in the frame. Because the dynamic range of the sensor is such that if you have direct sunlight, you just won't get any detail on the shadows, and it will not come out very well. So you always need some sort of shadow, and then it's a really good idea to bounce a little bit of light up onto the mushroom. So often what I'll do is I'll put a white t-shirt or a photography reflector or a white paper towel something that's brightly colored just outside of the frame. And that way it bounces up a lot of the natural light up onto the gills. And it's certainly possible to do that too much where it doesn't look natural, but you want just enough so you can see the upper stem and you can see the gills. It's not blacked out from a shadow, but also, it still looks natural. And it also helps a lot to have just a little bit of flash. So if you just flash the mushroom really hard to get a really harsh shadow from the flash, it'll look artificial. So what I try to do is have maybe 80% of the light be natural light and about 20% coming from the flash. And usually I'll do a handheld flash. So if the shutter is open for 2 seconds, 3 seconds, I'll flash the flash at really low levels from, maybe a dozen times while the shutter is open from different angles. Since you're flashing it a dozen times from different spots, you don't get any harsh shadows. And that really helps bring out the colors. It brings out the texture a lot, fills in all the spots that have shadows without making it look artificial. So if you do all that stuff, then you'll get a really nice picture of the mushroom. And that's really important for documenting it. And then it's really important to save the mushrooms for later. So you want to dig them out from the base. So you want to use a knife. And ideally, it's not a folding knife, because if you do much digging with one of those folding knives, the joint will break. So like a fixed knife is really good, and it kind of goes into a sheath. So you want to pop it out from below, especially with Amanita, but really with any mushrooms, the stem base is pretty important. So you pop it out from below, and then when I'm saving mushrooms, I'll put them in tackle boxes. So the tackle box is flat and has lots of different compartments, so it fits in my backpack, and that way all of the different collections stay separate. And if I'm collecting a lot of similar stuff, maybe a bunch of different Lacaria species in the same day, then what I'll do is I'll take a little piece of paper or a leaf or something, and when I'm taking the pictures, I'll write the photo number of one of the pictures down, and I'll put it into the compartment with the mushrooms, because later I want to know, which picture goes with which dried mushroom, and that's really critical. And after a few hours, it can get real hard to figure out, and after a few days, it's impossible, and after years, there's no way to figure out which mushroom went with which photo unless you write down the numbers as you take the picture. So when I get home at the end of the day, if I'm traveling, I'll put all of the mushrooms into paper bags and write the collection information on the paper bags. And it's really important to write the stuff that you can't see in the photo. So at least one of the photos needs to be taken with something for scale. It doesn't matter what it is, as long as it's something that you know the exact size of. Could be a ruler, could be a coin, just so you can figure out exactly how wide those stems were, how tall the mushrooms were, that sort of thing. Also, any odor or taste, it's really important to taste and smell all the mushrooms. And then also the trees that were around. And if your camera is not recording it, the GPS coordinates. So I write that all on the paper bag or on a piece of paper that goes in with the mushrooms. And then if I'm traveling, I'll just leave the mushrooms in the paper bag on my dashboard. And after a few days of driving around, they'll be really dry. So then I will feel, and if they're cracker dry, like they will snap instead of bend, then I know they're ready. And then I'll put them into Ziploc bags and write all the collection information on a slip of paper that goes into the Ziploc bags and put that into a larger Tupperware with desiccant. And the desiccant is important because these mushrooms will try to reabsorb humidity. So after a few months, you'll go back to study the mushroom and they'll be all moldy. So keeping them in Ziplocs with desiccant will keep them in good shape for hundreds of years.
That is the complete 101 on the best way to collect mushrooms for use in scientific study or make sure they're relevant to the furtherance of mycology. And I love your information about photography. I know there are people that love taking pictures of mushrooms, and you may have saved a lot of people 10 years off their life through your experimentation, your trials and errors, to now really share with us some of the really good insights about taking photographs of mushrooms. And it seems like you've probably had to develop your skills as a photographer a lot in exploring mushrooms. And obviously it is so important because as you're drying these specimens for storage, they don't look anything like those original fresh specimens. So getting a good picture at the time is absolutely critical. I also want to point out your dashboard trick that you taught me a couple years ago. I now use that for any forage I go on, put the mushrooms on the dashboard and they'll dry out. And I kind of share that one with everyone. So I'm glad you brought up the
Yeah,
dashboard
that works
trick
really
there.
well. And it's
Yeah.
also really important to make sure you upload your photos to Mushroom Observer or iNaturalist. I do both, so I upload all my photos to Mushroom Observer, and then there's an import script, so it copies all your observations from Mushroom Observer to iNaturalist. So you only have to upload them once to one site, and
Wow.
then you get them on both sites. So that's a really good way to do it. And then I label all the bags with the Mushroom Observer and or iNaturalist numbers. And that's one reason the iNaturalist app is really good, because if you take the pictures with the app instead of with your just your regular cell phone camera app, then at the end of the day, they automatically upload. You don't have to spend time when you could be on the microscope or just hanging out with your friends or doing other stuff at the end of the day. All that part is already done with you, and you can put collection notes and stuff like that in there. And also, a good thing to do with the mushrooms is to leave them in an herbarium. So a lot of universities have herbariums, and it's kind of like a library for mushrooms. So you can put your dried mushrooms in there. If you just have like some blurry photos and some broken up mushrooms, it probably won't want them. But if you have good collections that are well documented with good photos and a link to the iNaturalist number, that's really good for the herbarium, because most of these herbarium collections, they have descriptions, but they don't have photos, so they never really know what the mushrooms look like fresh. Herbarium curators are very happy to get collections that have good photos to go along with them, and they will save the mushrooms forever. So when a scientist is trying to study a mushroom, describe a new species, they'll go into the herbarium and study all the collections that are in there. So dropping it off at an herbarium is how you bridge the gap between the amateurs and the professionals and get the mushrooms into the professionals' hands, where they'll actually do the DNA sequencing and the careful informed microscopy that they can really turn into useful information.
Yeah. And with so much biodiversity, especially in kingdom fungi, the more people we have doing this, the better information. I mean, the more data there is for these academics to sort through and glean insights from. So it really is something that as more people start doing the process you're describing, the more breakthroughs we're going to see in the study of mycology, because there's just going to be more data, especially with something like biogeography, things like that. The more data, the better. So I'm trying to learn about these processes myself that you're talking about, integrate them into my own foraging. And I hope people listening will start to do the same, start to think of ways. Maybe it's just getting iNaturalist, taking photos, uploading them at the end of the day. There's easy ways you can start integrating this into your process. And I think there are some people out there that are going to love going through that entire process and feeling like you are doing some kind of scientific work just when you're out foraging. Especially I always kind of separate gourmet foragers from people that kind of forage for everything and are really interested in just the idea of fungal diversity. A lot of times they're one and the same, but it is great, even as someone who's more interested in gourmet foraging, to know that there is a process that'll make even those finds that people might call junk mushrooms, or basically not edible or medicinal, there's a way to make those still relevant and still give you a great forage, even if you don't find exactly where you were looking for. So I just want to get this information you just broke down so well out there to more and more people. And I do hope you start making some videos and showing people some of these tricks, especially when it comes to photography, because I think there are a lot of people out there that are obsessed with photographing mushrooms. I always say there's a large set of people that don't like eating mushrooms except with their eyes. So I think that kind of information would be hugely, hugely useful to people. And you just talked about that next step of microscopy and PCR. And as I understand, that's a lot of the current work you're doing. I know you just went on a tour teaching different mycology clubs around the country about PCR. So I know it's hard to sum up really quickly, but what is PCR and why is it important?
PCR stands for polymerase chain reaction, and it's a way to read the DNA that's in the mushrooms. So the mushrooms have maybe 40 million base pairs of DNA, so that's like a book with 40 million letters in it. And with PCR, you can cut out certain bits of those genes and then read them. So there's a lot of different ways to do that. The least expensive way is called Sanger sequencing. And Sanger sequencing allows you to read 1,000 of those 40 million bases for about $10. It's almost free compared to how cool the data is. So what we usually do is look for these parts of the genome that do absolutely nothing. And these spacer genes are unique to different species. So different species will have different spacer genes, and a closely related species will have a slightly different gene, whereas a distantly related one will have a very different gene. So by reading these sequences, you can barcode the mushrooms and really get a lot more insight into what it is that you found. So I extract the DNA, usually with sodium hydroxide and some other easy-to-get chemicals, and then run it through PCR, usually with ITS primers, because that's the best one for identification, and then with some other different primers to get some of the more conserved genes, if the goal is phylogeny, to figure out how they're all related. And then so you can see where exactly your mushroom fits in with the whole tree of life. That's real interesting because that's how you discover new species. And this gives you a whole much broader understanding of what it is that you're finding exactly. So the next time you see it, if it's something you sequenced and you're like, oh yeah, I know exactly what that is and what it's related to. And so the first thing you do with the sequence when you get it back is to go to NCBI BLAST, and it compares your DNA sequence with every other DNA sequence that's been uploaded to GenBank. And so you'll see a bunch of 100% matches if it's been found before. And the 100% matches show you where in the world this exact species occurs. And then you'll start seeing the 99, 98, 90% matches. So you can see what's closely related and where in the world those closely related species are. Then you can download all those sequences and calculate a phylogenetic tree. And it uses a bunch of math to figure out how everything probably evolved. So that gives you a lot of insight into the organism that we just didn't have 20 years ago.
And it's something now that has reached the point where amateurs and citizen scientists have access to this information more than ever before. Now, what is the simplest way for people to get a mushroom they found sequenced, get back that information? I believe, are there services that will take a dried part of that mushroom that hopefully you've kept, like Alan just outlined, hopefully you kept that dried mushroom. Are there services where you can send part of that dried mushroom and they'll do actually that PCR process for you?
Yeah, the one that most people use is called Alva Lab. And the website is alvalab.es, and they're in Spain. It costs about $20, and you just send them a little piece of a mushroom. They need a piece smaller than a grain of rice to get that DNA information out of it. And so, yeah, you can just send them all to them. And you can also set up a DNA lab in your house. And so it's not for everybody because, there's a lot of pipetting, a lot of mixing of chemicals and stuff, but it's also not very hard. It's more difficult than cooking eggs, but easier than baking bread. So I've gotten a lot of these labs set up in people's houses. And it's, depending on how much used equipment you want to get and how much you want to cut corners. Some people have done it for under $500. I usually tell people about $800 for a good robust setup with commercial equipment. Or if you buy everything new, it's probably more like $4,000. But nobody does that. Well, I guess there's some new PCR machines that are real small, and some people buy those new these days. PCR takes a lot of electricity because the machine is a thermocycler, which lowers and raises the temperature of the sample. So it's not something that you'll be running in the field unless you leave your car running the couple hours that the PCR machine takes. So usually you'll dry the mushrooms or just bring fresh mushrooms back home, back to the lab, and work on them there. And there's a lot of biohacker spaces now that are opening up. So regular hacker spaces, people break into computers and make electronics do things they were not designed to do, whereas a biohacker space, people will hack biological things, chemicals, figure out what all the microscopic features are and do the DNA sequencing. And they're like little community labs. They have them in pretty much most large cities. So you can just pay a few bucks a month and join these labs. I think the largest one is in Oakland, Counterculture Labs. There's certainly a lot of people doing mushroom stuff there. I know there's labs in Los Angeles, Seattle, pretty much all over the place. The advantage of this is that once you get a DNA sequence, you can upload your own sequences to GenBank. So anytime somebody else finds the same mushroom, they'll say, oh, wow, Alan found this a couple years ago, and they'll be able to see the exact coordinates that I found it. And they'll also be linked to my photos. So most of the sequences in GenBank are not linked to photos. So when you're doing this analysis, you don't know what it is that other people were finding exactly because you can't see it. But I always teach people to make these Mushroom Observer and iNaturalist records and then link their GenBank records to these citizen science website records So then when you get a match, you can see that it has photos, and you can see all of the other collection information and GPS coordinates and everything else that went along with that. That's one of the most important things that amateurs can do is just take good photos and then get DNA sequences and link their photos to the DNA sequences. And then, even 1,000 years later, somebody finds the same mushroom, your photos will come right up when they do that blast search. So it's a really good way to add to the knowledge of what's out there.
And it's a great way for amateur foragers to create a body of work all their own that continues on. And I think that's what everyone somewhere in themselves wants to feel, that they've put in this effort and this relationship with the forest and with the mushrooms, and now they've put it into something, some kind of body of work that will live on. Now, this is a huge question, but with the development of citizen science spaces like CCL there in Oakland, the increasing availability of the equipment, the increasing exponential growth in the knowledge of how to do these things on the citizen science level, simply because more people are doing them and are networked and sharing. Where do you see the field of citizen science in 5 or even 10 years? I mean, how advanced do you think citizen scientists are going to be? And what do you think their role is going to be in actually advancing the frontiers of science?
Well, one of the most important things is documenting where these mushrooms are. So if you can go onto these databases and just search for a species, you can see where in the world it's been found. And that's really important. And another really important thing is for conservation. If you're going to try to conserve an organism, if it doesn't have a name yet, it's very hard to conserve it. So all these rare species and very localized species need to have a name, so that way people that are managing forests know that there's something really special at this spot, and they can save it, because then they have an official name or a real reason to save some parts of the places that are very biodiverse.
And I just commend you for all the amazing work you've done in furthering citizen science. As a member of Counterculture Labs myself for a time, it was really such a huge resource to have you there giving talks, showing people how to run PCR. I mean, you were even making agar plates for the entire lab to use. So I just really appreciate all the contributions that you're making to citizen science and being willing to share your methodologies and how you do this. I mean, it's helped a lot of people further their own study and their own practice in such an amazing way. Is there any other work that you're doing currently, or anything else that we should know about that you're doing?
One thing I'm working on is like field guides. I'm not like sitting down writing them, but I go to the forest a lot and just try to get the nicest picture I can of every kind of mushroom that's out there and get DNA sequences attached to these pictures. So I'll be able to write field guides where each mushroom is identified not only by looking at it, but also with microscopy and DNA sequences, so people can see what they have. So I'm working on a field guide to Mexico that'll probably be out around 2030, and maybe one for California that'll be out around 2040.
Wow.
So that's kind of long-term stuff. Shorter term, I'm working on some posters. I'm working on a poster for the psilocybe of the United States and psilocybe of Australia. And for that, we're gathering the nicest pictures I can of all the different species and doing the graphic design work to make it into a large size, glossy, high resolution poster. And a lot of what I do now is just public speaking. So I try to give talks as often as I can. A lot of different mushroom clubs will fly me out. So I go all over the United States and Mexico. These mushroom clubs just fly me out, and I'll try to get people excited about mushroom photography and citizen science, and really nerding out on the stuff they're finding in the woods. And I think the main point of all of this is to really give people a deeper connection to nature, because everybody likes nature. But if you're really trying to identify the stuff that you find, really learn it on a deeper level, then you have much more of a reason to go out into nature and a much more of a reason to conserve it. So, anything that people can do to get people into mushrooms will be good, because picking mushrooms is not bad for the mushrooms. What is really bad for the mushrooms is habitat destruction. So whenever they chop down a forest to make a new shopping mall, a new condominium complex, those mushrooms will never come back. And so if we can get more people into foraging for edibles, then they'll see, a really good reason to keep the forest there because it's a sustainable source of food. Whereas if you chop down the forest to make farmer fields, that's another source of food. But if we can give people the knowledge they need to pick wild mushrooms, then they'll have a reason to keep these forests and have their sustainable sources of food there. The more we can get people into, really identifying the mushrooms and teaching other people how to identify the mushrooms, the more people will have more connection to nature and will be more interested in conservation of the habitats that we still have.
An incredible sentiment. And I think that is the first step in balancing our current human society's relationship with nature, is getting more people out into it and exploring it. And you said it so perfectly at the beginning that it's hard for some people to take a 4-hour walk in nature without a purpose. We're purpose driven in a lot of ways. So exploring kingdom fungi or just exploring biodiversity gives you such a great reason or vector to get into exploring nature and appreciating and communing with nature more and more. And I think that is such a huge step in pushing us toward society that's more in balance with nature, is getting out and communing with it and experiencing it. And everything you're describing is getting people there, whether they even realize it or not. The outcome is someone who's intimately more aware and connected with nature and wanting to conserve it. Now, we've been talking for just about an hour, probably over an hour at this point. I could listen to you talk forever. I hope we do get a chance to do, more podcasts and dive deeper even into some of these topics. But to wrap things up, I do usually ask people a few quick hit questions that I think give us an even deeper insight into who Alan is. So the first one of those is, what is a mushroom that we should know about that you love, and why? It doesn't have to be a favorite. It could change in 5 minutes. And I know for someone who knows so many fungi, it's kind of hard to pick. But what is a mushroom you love, and why should we know about it?
Oh, there's so many really unique mushrooms, but one that stands out to me is Lepiota trichroma. And this one was described about 20 years ago from the Mexican state of Veracruz. And I've also found it in Oaxaca. And it's really cool because it's bright yellow. And you find it in the cloud forest habitat. And then when you damage it, it turns bright green. It also has some kind of red flakes on it. So for that reason, trichroma. And so if you pinch the gills in that, you'll get a really bright green color. It's just like nothing else I've seen. So that's one that I always like to find.
Hard to beat brilliantly colored mushrooms and color changing mushrooms. Always a favorite. And then if you could speak to an 18-year-old Alan, given the experiences you've had and everything else, what advice would you give to your 18-year-old self?
Well, it really depends on what they want to do with mushrooms. Mycology is an extremely broad field, and I'm focused on a very small part of that, which is mushroom identification and photography and DNA sequencing. That's an extremely tiny part of mycology, and it's a part of mycology that there's almost no money in. And that's really good because there's no big corporations doing it. The people that get into it are into it because they really love nature, and they never expect to get rich or famous doing mushroom identification, mushroom photography. But, if somebody wants to make a living doing mushrooms, then there's certainly a lot of ways to do that. But if they want to get a conventional job, then they'll probably be either at a mushroom farm, which is kind of boring, repetitive work, but it makes money, or, plant pathology. There's a lot of fungi that attack plants. Now, they don't look very cool. They're kind of boring, but that's where all the money is. So if you want to make your living doing mycology, you got to figure out which part of mycology you want to do. And there's a lot of colleges that will have good mycology programs, especially San Francisco State, UC Berkeley, University of Tennessee, University of California, Humboldt, and University of Wisconsin, La Crosse, maybe Purdue. They can teach you a lot of stuff. So if you want to get a job in the field, probably need a degree. I did the opposite thing, where instead of getting a job in the field, I just take pictures and sell the photos to book publishers and do a lot of public speaking and teaching DNA classes and doing the DNA sequencing and all that kind of stuff. So for the stuff I do, there's really no reason to get a college degree because all of it's available online. But there's certainly a lot that you can learn that way too. There's a few people in the country that teach microscopy at a really advanced level, and that's something that is not super easy to pick up on your own. And You really won't learn mushroom identification very much in college. Maybe you'll take one class on it, but that's mostly learned on your own. But if there's an 18-year-old that wanted to really get into mushrooms and do what I do, I would say the most important thing is to spend a lot of time in the woods, and not just walking through the woods, but really paying close attention to the mushrooms. So when you see them, not only take good pictures of them, but pick them up and smell them, taste them, tear them apart to see the texture, the consistency, save them for later. Take pictures of the spores and all the microscopic features with the microscope, and set up a little DNA sequencing lab. And that's how you can really get into understanding these organisms. It's definitely something that more people ought to be doing.
Yeah. Get out there, show up, start doing the things you need to do to pursue this passion.
And of course, all the mushroom events too. There's a lot of really good mushroom events. There's a lot of fungus fairs. On the West Coast, the fungus fairs start in British Columbia up in August, and then just about every weekend, there's another mushroom fair to the south. So if you start in British Columbia and go to the first mushroom fair in August, you can just keep heading south and get the whole coast. The last mushroom fair is the middle of February in San Diego. All of the people that are really into mushrooms go to all these fairs. And then there's mushroom clubs in pretty much every major city, and they have a different speaker that comes in every week, so you can get to know all the mushroom people in your area and go to all these talks. That's a really good way to get plugged into the community as well. And there's also really good mushroom fairs in Mexico. There's one in Oaxaca the second week of July, which was canceled this year. It's there every other year. And that's in Coajimalollas, Oaxaca, just above the city of Oaxaca. That's a really cool fair. And then the middle of August, there's always the Sangu Fair in Michoacan. So that's not too far from Mexico City. You can just fly into Mexico City and head over there. That fair is a free event that's put on by the government, and they have lots and lots of tables full of wild mushrooms that are all identified. And so that's a really good one to go to. And there's mushroom events pretty much all over the place. And a lot of times you'll see like professors of mycology there at these events, and they'll bring all their students. A really good way to meet people and get more plugged into people that are doing things with mushrooms.
Network with your local mycology clubs, show up at mushroom events. Man, is it the fastest way to learn and meet new people, and even just find new inspirations for things you hadn't thought of. Like you said, mycology is such a vast world. And when you go to a mycology event, you start seeing all these different booths, all these people doing different things, and you definitely find some inspiration. And thank you for the inspiration for an epic road trip from British Columbia all the way down to San Diego. That one's definitely going on the list. That would be a pretty fantastic summer to winter excursion.
Yeah, maybe I'll do that this year. And if you really want to learn mushroom identification, you show up the day before the fungus fair, and that's when all the identification happens. So if you show up like during the fair, you can see all the mushrooms that are already identified, but to really talk to the people that are doing the identification and learn why they apply each name to each collection of mushrooms, you got to show up before it opens. That's when all the real work happens. Also just posting everything online is a huge thing, and just getting feedback from all the other people. And you post something on iNaturalist Mushroom Observer, and if you don't get the feedback that you wanted, then you have that link with the observation number, and you can send that to the different professionals. They all answer their email. So, there's always somebody that's studying a certain genus, and you can, if you don't get good answers from the people that check these sites regularly, then you can email the world experts and, get a professional opinion on that. And it's much better to link them to one of these sites where the information is public. That way you're not wasting their time just by sending them a bunch of pictures. It's more like their expertise is recorded forever. And so that's a really good way to dig into stuff as well.
That's great advice. And then the final question, with your whole body of work, everything you're putting out into the world, speaking, potentially field guides, everything else you're working on, what is the lasting impact that you hope to have?
I'm just trying to get people into nature more, get people to pay more attention to nature, and have a more thorough understanding of it, so they know like which parts of the forest need to be conserved, and give people a little bit more meaning in their life. Yeah. Because the working in an office job certainly pays the bills, but it can seem like you're not really making good use of your time a lot. So even if people just go off on the weekends and document nature, they can feel like their life has a whole lot more meaning. And I think that's the most important thing.
I always say mushrooms saved my life and saved me from an office job and had that exact effect. So I second that on and on and on. Well, Alan, thank you so much for your time and sharing all this information with us. I'm going to have to listen to this one again and take a lot of notes when I'm writing the summary and putting links together. But thank you so much for taking the time out and sharing your knowledge with us.
Thanks for having me on.